Hello, everyone. Thank you all on the other side of the line for attending this session in which I will be discussing the integrated solutions BioLegend provides for comprehensive characterization of antigen-specific T and B cells at the single cell level. I will first provide an overview of the innovative workflow and reagents that permit the simultaneous characterization of proteins and transcriptomes in single cells. I will then illustrate how these novel reagents are being used by scientists around the world to describe the phenotype, physiology, and function of antigen-specific T and B cells. And finally, I'll share exciting new products that we have recently released, along with information about an upcoming software release. To provide the best context leading to the development of this new technology, we have to first look at the technological landscape of cell analysis. One of the gold star techniques for cell analysis in the life sciences is flow cytometry. Now, flow cytometry is a ubiquitous technique used to characterize cells. Its development goes back to the 1950s when the culture principle was first described. In subsequent decades, commercial instruments were developed with more and more capabilities to measure emission signals and increasing number of detection channels. Modern instruments and advanced flow cytometry technologies now have the theoretical capacity to detect signal in dozens of channels. However, the number of available channels does not equal the number of parameters available to characterize cells. Technically speaking, what really defines a physiological parameter is the vehicle or reagent used to generate a signal that will be detected by a given channel in an instrument. For example, in typical flow cytometry experiments, we're measuring fluorescence emitted by a fluorophore conjugated to an antibody bound to a cell markers of interest. Until very recently, I think it's fair to say, that reagent availability was ahead of instrumentation. You can see in this graph that even just 10 years ago, the average number of fluorophores available was exceeding the average number of detection channels used in flow cytometry. Now, we all know that complex biological questions require more and more information to provide accurate answers. The need for higher resolution in dissecting overlapping cell populations or even discovery of new cell types has been driving the evolution of both more capable instruments and new dyes or fluorophores. Here to the right, I am showing you three common lasers that can be found in conventional flow cytometers: violet, green, and red lasers, along with the emission peaks or fluorophores that could be used to study complex cell populations, such as myeloid cells shown in the dot plots here to the left. However, as I mentioned a couple of slides ago, new technologies such as mass cytometry and spectral cytometry are now offering more channels than the number of available dyes used in multi-parameter panels that are used to characterize complex cell populations. However, even at this stage, protein detection still has an inherent limitation in which we can only detect a limited number of markers. With the advent of single-cell transcriptomics, cell analysis was transformed by the ability to characterize thousands of transcripts simultaneously at the single-cell level. The introduction of single-cell transcriptomics raises the question: can we combine the versatility and power of flow cytometry with the high throughput and granularity of single-cell RNA-seq? The answer is an emphatic yes. If we can measure sequencing reads for protein instead of measuring fluorescence, we resolve the limitations of flow cytometry by allowing the characterization of potentially a limitless number of parameters from a single cell. Measuring sequencing reads is done by replacing the fluorophore with an oligonucleotide that contains a PCR handle and a unique sequence barcode used to identify the antibody. Here's the structure of the initial antibody oligo-conjugates that were developed in collaboration with the New York Genome Center. These oligo-conjugated antibodies, or as we've termed them, TotalSeq antibodies, contain an oligonucleotide that contains a poly-A tail, allowing for the capture using poly-DT sequences. The oligo also contains a PCR handle compatible with the Illumina sequencing systems. Counts of bound antibody barcodes are determined post-sequencing by tertiary analysis packages. Since the introduction of the first TotalSeq antibodies, we have worked closely with 10x Genomics to develop reagents that are specifically compatible with their feature barcoding solution. We developed two families of reagents, TotalSeq B and TotalSeq C. This slide shows the structure of the TotalSeq C oligo. TotalSeq B and TotalSeq C antibodies are compatible with 10x Genomics 3' gene expression and 5' single-cell immune profiling kits, respectively. The major difference between TotalSeq B and C is the capture sequence that is employed. TotalSeq C antibodies are a key component in the experimental design of the data we will be discussing later in this presentation. The overall process is broken down into 5 major steps. Cells are stained with TotalSeq antibodies, then processed through single-cell partitioning, then sequencing, and finally analysis. During sequencing library preparation, 2 libraries are prepared: an antibody-derived tag, or ADT library, and a gene expression, or GEX library. Both libraries are sequenced, rendering the protein and transcriptomic data for subsequent analysis. This is the workflow as it was initially optimized and named CiteSeq by the researchers at the New York Genome Center. Now, moving on to the applications of these novel reagents, I wanted to start by describing the characterization of antigen-specific T and B cells, which couldn't be more relevant to COVID-19 related and other research efforts. Before going forward, I'd like to describe a tool based on soluble MHC molecules that can be used to detect antigen-specific T cells. Stable MHC tetramers can be generated by using a streptavidin molecule to bind 4 monobiotinylated MHC monomers. Each MHC monomer is bound to a peptide that is used to determine antigen specificity. The streptavidin MHC complex can bind an antigen-specific T cell receptor, or TCR. That MHC construct is often called an MHC monomer, even when technically it is made up of 4 individual molecules, the 2 MHC chains, a specific peptide, and the biotin molecule. Now, BioLegend provides a superior and extremely flexible version of this tool called FlexT that is based on the utilization of ultraviolet light sensitive peptides. The peptide of interest is incubated with the MHC monomer and placed under an ultraviolet light source. The UV light sensitive peptide that has been sitting in the MHC molecule is degraded, allowing the relevant peptide of interest to bind the empty MHC monomer, stabilizing the complex. Tetramers can now be constructed using the MHC monomers loaded with the peptide of interest. This can be repeated multiple times, every time you need a new tetramer, in fact. If, in addition to the fluorophore, we now add an oligonucleotide, we have an MHC tetramer that is now compatible with single-cell RNA sequencing and immune profiling via sequencing. In the next few slides, I'll be sharing the results of experiments using TotalSeq antibodies and FlexT tetramers. In the first set of experiments, we stayed PBMCs with a fluorescently barcoded tetramer loaded with cytomegalovirus, or CMV peptide, and a TotalSeq antibody panel that contains markers used to identify basic immune cell types, such as T cells, B cells, NK cells, and monocytes. Here to the right, you can see how well the flow cytometry data correlates with the CiteSeq data. Note that BC indicates barcode. Taking advantage of the power of single-cell analysis, we're able to project cells into clusters based on antibody detection. The plots shown here, called UMAPs, or Uniform Manifold Approximation and Projections, show the relative relationships of cells based on their expression levels of certain proteins, as measured by TotalSeq antibody staining. Each plot is associated with a specific marker shown on top of the plot. Expression levels are denoted by a color range, where dark purple or green indicates higher expression of a given marker. You can see the cell clusters as defined by the basic surface markers: B cells in the upper left plot, T cells next to it, followed by CD16 positive cells, CD4 T cells, and myeloid cells in the upper row. Down in the bottom right panel, we can clearly see the CMV positive tetramer population, with the green circle following this positive population through the individual UMAPs shown on this slide. The quantification of the different TCR clonotypes was done with Cell Ranger following the single-cell immune profiling workflow, and you can see how we can identify the reactive clonotypes from the CMV stained and processed PBMCs. Moving on to B-cell characterization, I'm going to show some data from our collaborator, Patrick Wilson from the University of Chicago. But before diving into the data, I want to illustrate how they can make a similar tetramer using our fluorescent and barcoded streptavidin. Working with influenza virus, they monobiotinylate the hemagglutinin antigen, which is one of the immunodominant antigens of the flu virus. With that biotinylated antigen, they can now make a tetramer which will specifically bind to B cells that express surface receptors that recognize this antigen. These B cell receptors are immunoglobulins that recognize the antigen and thus the virus. For many years, researchers have been looking for a universal flu vaccine. The best candidate will be one that confers broad protection and has potential to be quickly adjusted to treat pandemic outbreaks. One strategy that's been followed is to stimulate production of antibodies that recognize the stem of the HA or hemagglutinin, which is a more conserved section of the antigen, compared to the highly mutagenic head. To achieve that, immunizations can be done with the headless stem, as indicated in the upper panel of the figure, or with chimeric antigens containing exotic heads that are not commonly found in strains that infect humans, as illustrated here in the bottom panel. The goal of this project is to try to construct one such universal vaccine candidate and also isolate a neutralizing therapeutic antibody. To accomplish this, the Wilson lab analyzes memory B cells and also baits them with an array of tetrameric barcoded antigens. On the left hand, we have sorted memory B cells as defined by their surface phenotype, which is CD19 positive, IgM low, CD27 positive, and CD38 low. On the right-hand side, experiments are done with the baited B cells, in which we can detect antigen-specific B cells. To further characterize cells isolated, the Wilson lab used a panel of 32 different TotalSeq antibodies, which they titrated shown in the table to the left. After sorting the memory B cells, they can identify 7 different clusters of cells using both protein and RNA in an integrated analysis. As you can see here, antigen-specific B cells represent the large cluster to the bottom of the t-SNE plot in red color to the right. Here again, you can see the correlation between the flow cytometry characterization during the sorting step and the expression of the same markers in the t-SNE plots, showing high expression for CD19, CD27, but low expression for CD38. The use of additional markers allows for deeper characterization and also internal validation. As expected from memory-activated cells, they also show high levels of CD44. Below is the full heat map of proteins and RNA expression for the 7 clusters. May be a little difficult to see, but on the top they annotate the expression of proteins. The combined analysis of protein and RNA and its correlation can further refine what information is more useful in a particular setting. For example, here they can quickly eliminate which genes or antibodies may not be that informative, as they can't be detected in their cells of interest. Those are the ones in the grayed out area. And when looking at the rest, it is apparent that a combination of protein and mRNA expression may be the best biomarker landscape for their cells. Now, in these sets of experiments, they are looking at individuals that have been vaccinated with 2 chimeric vaccine candidates. The B cells are sorted, baited with tetrameric antigens. When looking at the mature B cells characterized by expression of IgG and IgA, annotated in light blue here in the heat map, they can see a larger proportion of the B cells that were FISH with the chimeric tetramer, meaning they recognize a large number of viral strains. They also see a larger expansion of V locus VH1-69, which is a clonotype that has been described as highly cross-reactive across multiple flu strains. In this final data slide for this project, you can see the correlation between the barcode read count to the left and the affinity of the antibody produced by the corresponding B cell to the right. After analyzing the single cell data from the antigen-specific B cells, they cloned the B cell candidates based on their cross-reactivity and expressed the antibodies. The ELISA was done coating the plates with the antigens as indicated at the bottom of the graph. The cloned B cells correspond to the antibody expressed by raw counts between the two graphs. Okay, so moving on. Now I will introduce two new products and an upcoming software release. The first product is the TotalSeq Human Universal Cocktail version 1.0. Followed up by that, I'd like to quickly introduce two new recombinant SARS-CoV-2 S proteins. And finally, I would like to introduce our upcoming software release called Multiomics Analysis software. Okay, so the first product is the TotalSeq Human Universal Cocktail. This cocktail contains 130 pre-titrated lyophilized primary antibodies plus 7 isotype control antibodies and is compatible with 10x Genomics Chromium single-cell immune profiling solution. The Human Universal Cocktail is the culmination of a years-long program aimed at creating the first lyophilized antibody cocktail for use in single-cell experiments. In the next few slides, I will review the process by which this cocktail was produced. So just a few words on how this cocktail is used. The universal cocktail can be easily integrated into the 10x Genomics 5' single-cell immune profiling workflow. By doing so, users can obtain TCR and/or BCR sequences, gene expression, and protein expression data from a single cell. Before showing some data for this cocktail, I'd like to review how this and future cocktails are built. Cocktails move from conceptualization to creation as an unoptimized formulation, meaning that the optimal antibody concentrations have not yet been determined. The unoptimized cocktail undergoes rigorous titration testing using primary cells and cell lines under various simulation conditions. Antibodies that provide good, positive, and negative cell discrimination are kept in the formulation for further testing. Antibodies that demonstrate poor discrimination may undergo alternate testing conditions, for example, a different cell line or activation condition than initially employed. If performance cannot be recovered, the antibody is excluded from the cocktail. Once a cocktail's contents have been selected and adjusted, the cocktail undergoes further testing and may result in additional adjustments. Once a final formulation is determined, we manufacture a pilot lot and perform final testing prior to release. In the previous slide, I mentioned optimal discrimination of positive and negative population. What this means is that we identify the antibody staining concentration that yields enough separation of positive and negative cells with the fewest sequencing reads. This is important to note because in other antibody staining applications like flow cytometry, we aim to maximize the signal-to-noise ratio of fluor-conjugated antibodies to their isotype controls. The consequence of this approach, when applied to single-cell protein analysis, is that you end up increasing consumption of sequencing reads, which are a limiting resource. This process is fairly straightforward. However, it becomes more challenging as we scale up and start including more and more antibodies in these cocktails. So the importance of titrating a large number of antibodies cannot be understated. On the left-hand column, we see the results of a staining experiment with an unoptimized concentration of anti-CD54. A total of 8.5% of pass-filter sequencing reads are attributed to this antibody. On the right-hand column, we see the results of a subsequent staining experiment with a panel containing an optimized concentration of anti-CD4. In this case, we're able to reduce the percentage of sequencing reads attributed to this antibody by approximately 10-fold, while still obtaining good discrimination. The result of this rigorous development process is a robust solution that can be used for multi-omic characterization of single cells. On this slide, I'm showing a small subset of immunologically relevant protein markers and their corresponding mRNA expression levels. This cocktail enables the detection of 130 protein surface markers. We can use these data to define immune cell subsets and create cell legends based on protein expression to aid in navigating such highly dimensional data sets. Delving a bit further into our high dimensionality dataset, we can begin to explore the cellular heterogeneity of a given sample. In this slide, I am showing the presence of different CD8 positive T cell subpopulations based on protein expression. In the first column, I'm showing expression of CD8 and CD4 to help us orient ourselves, top and bottom respectively. We can visualize distinct cellular populations based on expression patterns projected onto the UMAP shown at the top of the slide. Based on expression of specific markers, we can delineate the presence of naive, central memory, effector memory, gamma delta, and mucosa-associated invariant T cells, which are found at low frequency in PBMCs. Now keep in mind, this is just data showing the expression of a single major subset of immune cells. The next product I would like to introduce is the recombinant monobiotinylated SARS-CoV-2 spike protein, which we have available in two formats. The first format is the S1 receptor binding domain from arginine 319 to phenylalanine 541, and the second is S1 protein from valine 16 to arginine 685. Both of these proteins are monobiotinylated and can be used to create antigen tetramers using the TotalSeq PE streptavidin. And finally, I would like to introduce our upcoming software release, Multiomics Analysis software, which is a free cloud-based program for the analysis of single-cell data. This program is geared at bridging the divide between single-cell and flow cytometry analysis and is meant to be accessible to users of all backgrounds. It enables demultiplexing cell hashing data and takes outputs from Cell Ranger and Seurat as inputs. MAS provides an easy-to-use and navigate GUI. On this slide, I'm showing the demultiplexing page of MAS, where users are provided with demultiplexing results and basic data cleanup metrics. MAS also offers users the ability to define populations based on gating using protein expression data. Gating defined cell populations can be projected onto common dimensionality reduction plots like UMAPs or t-SNE plots. In this example, Tregs were defined as CD3 positive, CD4 positive, CD127 low, and CD25 high. By using MAS, I was able to show that these cells also express FOXP3, which we all know is a transcription factor for Tregs. We expect to have this software available by the end of the year. Okay, so in conclusion, the use of oligo-conjugated antibodies with RNA and TCR or BCR profiling provides an unmatched characterization of these cells at the single-cell level. This encompasses unprecedented protein characterization with TotalSeq antibodies, better definition of cell states with a combination of RNA and protein expression, as well as T or B cell receptor sequencing correlated with antigen recognition. BioLegend is committed to advancing the field of single-cell multiomics by providing easy-to-use and cost-effective antibody cocktails, along with novel reagents to aid in the study of immune responses to SARS-CoV-2. Finally, with the release of MAS, BioLegend is committed to not only providing reagents for single-cell multiomics, but also software to aid researchers in exploring high-dimensionality datasets. You can find much more information on our TotalSeq reagents and applications on the BioLegend TotalSeq webpage. Thank you for your time.